Welcome to the video that'll introduce the brain, and we'll get into some of the basic anatomy of the brain in this video. Now, not surprisingly, there's going to be a lot of language as we go through this chapter. Remember, first year university is often about acquiring the words that we're going to need and the basic concepts. So let's start with just a couple of the directional terms that we can think about applying to the brain. And one is rostral. So rostral is towards the forehead. And caudal is a term we'll use to refer to towards the spinal cord. And we'll see as we start looking at the brain, there's going to be a number of features. We're going to see, there's some landmarks like the longitudinal fissure, which we'll have a look at briefly. It separates the cerebral hemispheres. There's also structures, which we call gyri, which are these folds, and in between the folds are grooves, which are sulci. And then inside the brain, there'll be a few major landmarks that we can easily see, such as the corpus callosum. Now, before we go too far, just want to talk about the brain. The brain is unlike the heart, in that if you do a dissection of the heart, you can see structures quite easily, and the structures really give a sense as to the function. The brain is not that way, because of course we're dealing in a world of neurons, neural nets, neurons talking to each other at various centers communicating. So we don't have quite the same visual sense of form and function. Now, the brain we can look at in terms of some really major portions. The cerebrum, which is made of the cerebral hemispheres, that makes up quite a bit, about 83% of the brain volume. And here we can see the gyri, the sulci, the longitudinal fissures, corpus callosum. Towards the back and lower down, we have the cerebellum, and it's the second largest region. And this is actually a very important region because it has a really high density of neurons, about 50% of the neurons there, even though it only makes up about 10% of the brain volume. And it's nestled into sort of a little bony cavity within the skull, which is the posterior cranial fossa, which we'll have a look at next semester when we will look at osteology, or the study of bones. And then the brain stem includes different regions, such as the diencephalon, the midbrain, the pons, and the medulla. Now, just have a quick look here. We can see a few of the features. This is looking down on the brain. We can see the longitudinal fissure, which runs between the two hemispheres. And of course, we have the two hemispheres. We can also get a good view here of the gyri. The gyri are these folds, and you can see that there's a number of gyri. It's one of the big hallmarks of the anatomy of the brain. And in between are the grooves, which are the sulci, or the sulcus for individual. Now let's have a look from the side. We can see the major parts of the brain. We can see the cerebral hemispheres outlined here. We have the cerebellum. And we can see part of the brain stem here, such as the pons, the medulla, etc. Now, part of the brain stem is buried deep within here. Now, we have, as I said, we do have these things, the gyri and the sulci. And there are a few landmarks that, if you look carefully, you can start to notice. And I'll point at one of them right now, which is the central sulcus. And here's the central sulcus. You can see it running down. And this is going to be important to landmark different regions, because what it does is it actually separates a major motor and major sensory area, control area. And that's the central sulcus. Here's another one we could have a look at, and this is the lateral sulcus. So not all sulci have names, but there's a few that are very distinctive and separate major areas. You can see here that we have the temporal lobe, which is a portion of the brain. Now, looking inside, here we've done a mid-sagittal section, and we can start to see some structures. We can see down here we have the brain stem. Okay, we're down here, the brain stem. And we can see that the first bit is the medulla oblongata. Then we have the pons, and then just above that we have the area, the midbrain. Here we have the corpus callosum, and we're going to see, though, that's really a group of fibers. It's a big, huge, thick fiber tract that connects the right hemisphere with the left hemisphere and vice versa. So these are tracks that are going to be traveling right to left, left to right in the higher part of the brain. We also can see that we have the cerebellum here. And inside the cerebellum, you can see some distinct characteristics. You can see this white fiber track. It almost looks like a tree if you look at it, that all these white fiber tracks. So much so that it actually receives the name Arbor Vitae, which refers to the tree of life. Now this is a mid-sagittal section, so it means we're exactly separating the right from the left equally. So we're right down that midline. And here is where you can actually start to see some of the ventricular system. Now we can't see the lateral ventricles because they're actually buried within the hemispheres. So they're off to the side from mid-center or mid-sagittal. But we can see some structures. Here is a thalamus, the thalami. And hopefully you've watched the 3D brain anatomy video that I posted. And that'll give you a better sense as to what the thalami look like and their location relative to other tissues. If you haven't watched that video, make sure you go watch it. Probably a few times would be very helpful. We can also start to see some of the ventricular system. As I said, we can't see the lateral ventricles because there's one buried in each hemisphere. But we can see things such as the cerebral aqueduct, which carries fluid from the third ventricle. The cerebral aqueduct carries it down to the area of the fourth ventricle, and you can see this is midline between the brainstem and the cerebellum. Now, I just want to say a few things. Here's my, very, minimalist drawing of the view we just saw. But just want to put a few things in perspective. That when we're looking at gray versus white matter, we saw in the spinal cord that the gray matter was deep and the white matter was more superficial in a cross-section of the spinal cord. Here it's a bit different, is that we actually have the gray matter, and that's what I'm putting here. The gray matter is towards the periphery, so it's superficial. And so that's where much of the gray matter is. It's right in that outer layer of the brain. And then there's islands of gray matter deep within the brain. But beyond that, much of what we see in the brain is actually basically white matter. Now, this white matter is, of course, made of myelinated fibers traveling. And if you think of the functionality of the brain really is about communication, especially different areas. And not surprisingly, that what we have are a number of fibers. So we certainly have fibers coming up into the brain. So a lot of those white fiber tracks are about fibers coming up and being distributed, or fibers going down also. We also have fibers, fiber tracks that go from the front part of the brain to the posterior part. So that also makes up a lot of these big white fiber tracks. So we have fibers going into the brain, out of the brain, which is basically up and down. We have fibers going from the front to the back. And then, not surprisingly, we have fibers that are going to go from one side of the brain to the other. So we'd have a third set of fibers that are going to travel basically from one side of the brain to the other. So just to restate that, a lot of the gray matter is in the periphery. There's deep islands of gray matter within the brain. And then much of the brain is white fiber tracks of fibers going either in or out of the brain, going from front regions of the brain to the back regions of the brain, or from one side of the brain to the next. Now, we can just see this we can just look at some of the view again, in that we can see this in a real human brain. And you can see, things such as the cerebellum. Once again, you can see the medulla, the pons. We're into the midbrain, the thalamus, corpus callosum. Now, as I said, we have the gray matter. And the gray matter, it's basically cell bodies, dendrites and synapses. And why is it that color? Because we really do not have a whole lot of myelin here. And it forms basically the surface layer, the cortex of both the cerebrum and cerebellum. And as I said, there's islands of it deep within the brain. And what are these islands of deep gray matter? They're nuclei. And remember, nuclei are basically collections of cell bodies in the central nervous system. And then we have all the white matter, and they're the bundles of axons. And because we have lots of myelin, that's why they look that way. Now, we call them tracts in the CNS, kind of equivalent to the term nerves in the PNS. And as I said, we find these deeper within the brain. Now, just a little bit, we have to mention a little bit about the formation of the nervous system. This is not an embryology course, but we should mention a few things. But really, the nervous system develops from the ectoderm. And if you remember to previous discussions, at a certain point during development, you have three layers. You have the ectoderm, the endoderm, and the mesoderm in between. And by the time you get to that stage, each of those layers will take on a responsibility to become certain tissues. And here for the nervous system, it's the ectoderm we're going to focus in on. Think of it as sort of a big flat area of cells. And what happens is, right in the midline, you start to get a thickening of this layer of cells. And we're going to give it a special name. It's going to be known as the neural plate. Now, the neural plate's going to keep thickening, and as it does, it's actually going to start to sink down. And we'll see a diagram. And it sinks down so much that you actually get these things. It's going to be called the neural groove. And on the sides become the neural folds. And eventually the neural folds will come up and meet each other. And I end up with a hollow tube. And that tube is what's going to become the tissue that makes up the tubes. Think of it as a drinking straw. Sort of a look to it. The material that makes up the tubes will become the actual brain and spinal cord, and also the central canal will be that empty space or the lumen within that tube. Now let's just have a look. So here we are. Believe it or not, that's what we all looked like at one point. And we're looking at the top layer. And the top layer, you can see how it's starting to thicken in this area, and that's the neural plate that's starting to form. And you can also see how the neural plate is starting to sort of drop down. Now that's going to continue. It continues quite rapidly, and you can see a day later that the plate has actually thickened quite a bit, and it really has dropped down. And as it's done that, you can see the rest of the surface has sort of stayed up here. And that forms these folds that are going to run longitudinally along the top surface of the embryo. And so we have the neural groove, which is a space. We have the neural folds. And at the very top of the neural folds, this group of yellowish colored cells that they're trying to illustrate here are the neural crest, or the neural crest cells. Now this is going to keep developing, and you can see that the folds are going to move towards each other. Think of it almost like a zipper, where you're going to come along and zip it up. That these edges are eventually going to meet, and it's going to zip itself along over a period of time. Now, once that whole process has happened, I've now developed a neural tube. And that neural tube, of course, will run the whole length underneath the surface. This tissue here is what's going to eventually turn into the brain and spinal cord. Inside here, this empty lumen is going to basically become part of the ventricular system and the central canal that runs along the spinal cord. And that group of cells that form the top part of the folds sort of breaks off and stays here. And these become the neural crest cells. And they have a certain job, what they're going to do. They're going to become a certain number of items over time. So the neural crest, and that's that longitudinal column on each side of the neural tube. And of course, it came from ectoderm. And those neural crest cells are going to become part of the two inner meninges. They contribute to the peripheral nervous system and other structures such as skeletal, integumentary, and endocrine. Now, so we've now developed a tube, So we now have this tube. Now we have to try and turn it into what we now recognize as a brain. And we're going to see that there's going to be some modification of it. I almost think of it, if you've ever seen someone who makes balloon animals, that's what they're going to do, is they're going to take a long balloon and start to turn and twist, and bits will pop out and bulge out. And that's how we're going to make the brain. That's the short answer. And we're going to create a forebrain, a midbrain, and a hindbrain, also known as the proencephalon, mesencephalon, and the rhombencephalon. Now, we're not going to get too hung up about all the terminology when it comes to the embryology and the folding and the derivations within the brain. So let's have a quick look. So what we've done is we had a tube, and you can see I've drawn the tube over here. And what happens is part of the tube will start to bulge out. And we can see sometimes it really bulges out quite a bit. And over time, as that tube starts to change, you can see how its shape has really changed. So it's gone from a simple tube to, we got a bulging area called the rhombencephalon, another bulging area called the mesencephalon, and the top bit, which is the prosencephalon. And over time, we're not going to get too hung up about a lot of these terms, is you can see that the tube keeps basically changing. That here it almost looks like it's starting to develop Mickey Mouse ears, okay? And that's a bulging out. Now, if you start to look at it and think about the final structure, you start to get the sense that this is starting to look maybe a bit like cerebral hemispheres starting to form. We can look here that this bulge, you can see how it bulges out, really bulges out, and it does this. Well, not surprisingly, this is going to become part of the eye structure, basically contribute towards the retina, which is the neural component of the eye. And if you want to, you can really start to look at the comparison here as to the color coding of the structures and what it looks like in the embryo, and the color code in the structures, and eventually what it eventually becomes. So you can see that the bulges up here did indeed become really part of the cerebral hemisphere. You can see here part of what's, the diencephalon. Basically, it bulges out and becomes part of the eyes, and also becomes part of the ventricular system deep within the brain. And then, the brain stem from these tissues. So let's just talk a bit about the meninges. They're the three membranes that surround the brain and spinal cord, and they help to separate the nervous tissue from bone. And it acts as protection and support for the brain. And we've seen in the spinal cord that the meninges were there. And we've seen there that we have the dura, the arachnoid, and the pia. So it's very similar. So not only does it provide some protection, we're going to see it also has a role in terms of sort of that water layer that bathes the brain, called cerebrospinal fluid, has a role in its production and also circulation and return to the blood. And also provides a structural framework for the arteries and veins so they can actually travel around. Now when I look at the dura, remember the dura is the outermost layer. Tough mother is what it means. I have two layers. I have the outer periosteal. Now this is a bit of a clue right here, osteal. Osteo refers to bone. So it's very equivalent to something that we're going to see next semester called the periosteum of bone. Then I have the inner meningeal layer. And this continues into the vertebral canal and forms a dural sheath around the spinal cord. Now, there are a few places where these two layers are actually separated. Most of the time, they're actually together, but there's a few places where there's actually a gap between these, and we'll have a look at those. Now, the dura is pressed right up against the bone, and there's no epidural space. Unlike when we saw in the spinal cord, we had that fat layer between the dura and the bones of the vertebrae. We don't see that here. It's just basically dura is right up against the inside of the skull. It's not directly attached to the bone. There are a few places like the foramen magnum, which is the big hole in the bottom of the skull, the stella, the crista, and sutures. Now we haven't seen those structures, so we're not going to worry about them too much. As long as you know that it is attached in a few places, but most places it is not. Now, there are a few places where the dura mater actually extends in between sort of spaces that the actual brain has left. So part of the dura actually dips down between the cerebral hemispheres in the region where the cerebellum and the cerebrum meet. There's sort of a little space where it sort of wedges itself in there. So there's little areas where the dura wedges itself in. And they take on special names. Like the falx cerebri separates the two cerebral hemispheres. The tentorium cerebelli separates the cerebrum from the cerebellum. The falx cerebelli separates the right and left halves of the cerebellum. Now, the meninges, we have the arachnoid. Now, as we're working our way further, or deeper, I should say, we've gone from the dura to the arachnoid. And it's a transparent membrane. It's fairly transparent. And below the arachnoid, there is a space. So between the arachnoid and the pia, we'll see there is a space. The pia is very thin. It's almost impossible to really see it with your eyes. Because it's almost like a shrunk wrap on the outside of the brain. It so closely adheres, and it follows all the contours of the brain. It follows the arteries as they penetrate into the cerebrum. Let's have a look here at the meninges. You can see here is the surface of the brain, and here is the pia. The pia is really adheres, so every nook and cranny the pia is attached on and covers that surface. So that's the pia, that's the deepest layer. Then you notice that there is a space, there's actual space between the pia and the more superficial one, which is the arachnoid. And there you can see there's the arachnoid. So we have from deep superficial, the pia. Then I have a space, which is called the subarachnoid space. Then I have the arachnoid. And then right above the arachnoid is the dura. And so this is the dura right there. And as we look carefully, we can see the dura actually does have two layers. You have the periosteal layer, which is the outer layer right underneath the bone. And then you have the meningeal layer, which is deeper. And as I said, most of the time, there's no gap between them. You can see that they're continuous. They're continuous. But there are a few spots where they split open, and here's one spot, and it's called the superior sagittal sinus. And the name tells you a lot. Superior means up top, sagittal means in the middle, from front to back, and its sinus is a gap. And you can actually see that here the periosteal layer and the meningeal layer have actually split, and this provides a blood sinus. Now, why would I want a blood sinus there? Because in the next video, what we're going to see is when we make CSF, cerebral spinal fluid, it is made within the ventricles. It circulates through the ventricles. Then it circulates, much of it circulates on the outside of the brain in the subarachnoid space. Now, because it came from the blood, and it's not exactly the same as blood, but it's a component of blood, it gets returned to the blood, and it gets returned to the blood. Here's one, you see it right here, an arachnoid granulation. It gets returned to the blood through these special structures, the arachnoid granulations. And so this sagittal sinus here, this venous sagittal sinus, collects basically the CSF that has flowed through the brain. And you can see here, I'm going to try and zoom in a little bit more, that the arachnoid layer forms these, it almost looks like a little broccoli or a cauliflower right there. You can see another one over here. Forms these little structures, and in these structures what happens is the CSF will basically, the fluid will re-enter into the blood system. And we're going to give these a special name. They're known as the arachnoid granulations. And are those things right there. And we'll talk a bit more about them. Here you can see one of those places where the dura actually wedges itself in between gaps in the brain. And here's an example of the falx cerebri. And you can see how the dura just comes down here and really kind of gets into that groove right there. Okay, so I'm going to mention a bit about meningitis. It's Remember itis. Itis is an inflammation. So this is an inflammation of the meninges. Serious disease, especially infancy, childhood. Now, meningitis could be both either bacterial or viral. And often the route of entry is through the nose or the throat. And often it's the pia and or arachnoid that are most affected. And of course, as soon as you get inflammation in the brain, it's a closed container. You can build up pressure. So it can cause swelling of the brain, enlargement of the ventricles, and may actually result in hemorrhage. Typically, you often see high fever, stiff neck, drowsiness, intense headache, and of course, sadly, it can often progress to coma or death within hours of onset, and it's often diagnosed by checking out the cerebral spinal fluid through a lumbar puncture, most people know as a spinal tap. Okay, we're going to end this video here, and we're going to come back and talk about the ventricles and CSF in the next video.